Awesome. Cool, welcome back. Super excited for our afternoon session about EEG notebooks. Before I let Kevin and John take over, we'll have amazing stuff to show us. I kind of wanted to plug the Zoom call that I'm having for the data carpentry project, which will be happening in parallel to this, but that'll be M or Python based, and this is super exciting with EEG notebooks. But I'll put the information about that Zoom call on the Discord and the Mattermost channels. For those of you who have actually been looking at the data carpentry notebooks and want to give us some feedback. If not, you can also give us the feedback on Discord or Mattermost, or in the issues of the project. So with that, I'm going to log off and let Kevin and John take over with the excitement of EEG.
Okay, I'll crack on. So welcome everyone to the afternoon session, and the plan for the next 1.5 hours of the program is for me to be telling you some fun things about this project that I've been involved in for a while. The way this is going to go is, actually I have this in a slide, so let's just move on. By the way, the enigmatic and exciting figure that you're seeing right now will be explained later on if it's not super clear already. Okay, so this is going to be an introduction to EEG notebooks. This is going to be a 2-part session. The first part is going to be a kind of slide-based lecture overview background, me and some slides, and you guys watching on the Crowdcast. And then the second part is going to be a part live demo tutorial where I'm going to give some examples and go through some of the tutorials that we have for the tool, and kind of expand on the document, on the notes and comments that are in the documentation there already. So we'll do the first part, then we'll have a break. I'm going to aim for depending on how we're doing for time, either a 10 or a 15-minute break, and then we'll resume for the second, and that'll take us up to 3 o'clock. Okay, so let us proceed. So here we go. Part 1, overview and background. I'm John, in case any of you don't know me by now. The plan for this session then, so the first half of our 2-part thing, is first I'm going to give you a bit of background in terms of just motivations for why we would be interested, why we're putting some efforts into developing open source software for doing cognitive neuroscience experiments with mobile EEG, and then do a bit more of a deep dive on the actual tool, on the actual library, like what it is we're doing, how we're doing it, some of the implementation, all aspects, and some more examples of what it's all about. Okay, so top level, really, motivations. I mean, why? The first thing to ask is, why do EEG? Right? What's the benefit? This is largely a neuroimaging crowd, so a lot of this is probably familiar to people, but that not necessarily everyone. So the things that you tend to see, say, in first page of a lot of neuroimaging cognitive neuroscience textbooks is the following. So EEG is generally understood to be a direct measure of neural activity. So this is generally, we contrast this typically with fMRI, which is a indirect signal that is generated by metabolic processes. Just trying to increase my laser pointer. That's what I want. Okay. So it's good to be measuring neural activity directly. Again, in contrast to fMRI, like we don't understand a lot about what those metabolic aspects of hemodynamic signals are, and they have their own complexities. So it's nice to be measuring neural activity directly. The key thing, really, that is generally highlighted as an advantage of EEGs is temporal resolution. Right, so you're really measuring neural activity on the time scale of neural activity, as in on the order of milliseconds to tens of milliseconds, right? So that's, when you think about what is like the characteristic timescale at which neurons function, it's really on the order of milliseconds, really, in terms of like generating action potential and having a neuronal response to an input. And then at the aggregate level, neural populations, they tend to fluctuate on timescales going from on the order of tens to hundreds of milliseconds. So, we can see that with EEG. We can crank up the temporal resolution as high as we like. We can crank it up to 4 kilohertz and completely oversample what we need. But really what we need is a roughly, let's say, up to 1 kilohertz sampling rate for some fast signals and slightly slower than that for other signals, but it's all fast temporal resolution. And the two things that we get out of that, having a fast temporal resolution imaging modality, is firstly, we can actually watch neural activity evolve or watch the neural processes underlying cognitive functions evolve as they do on the, typically in that case, we think about on the order of hundreds of milliseconds. And the canonical example of that is called the ERP, or the event-related potential. And so this is the kind of thing that you're normally thinking about with EEG or with cognitive neuroscience type EEG experiments. You have these waveforms that have these inflections, we call, or these components at certain characteristic time points, like 100 milliseconds, 200 milliseconds, 300 milliseconds, and they have this, wavy shape. And these are understood to be aspects of brain responses that, say, index different components of the good processes that are underlying, say, the visual perception. Okay, so we can see the evolution of neurocognitive functioning on the time scale that it evolves. That's one of the features of having good temporal resolution. The second feature is that we can see oscillatory activity. If there's one thing I think that you could say, what is the brain? What does the brain do? I mean, there's multiple answers to that question, but one very, very good answer to that question is it's an oscillatory engine. It generates oscillations everywhere, all the time, multiple frequencies, interesting complex dynamics that are, that are going on at multiple spatial and temporal scales, but oscillations are everywhere, and particularly oscillations happen within the range of 1 to 100 or 0 to 100 hertz, and so that's something that you really need to be seeing. You need to be sampling and have fine temporal resolution for. So we see that with EEG, and that's a real feature of it. In terms of these other advantages, I mean, EEG's been around a long time, so well understood. It's compatible with other neuroscience, or let's say neuroimaging brain measurement modalities. This is quite an important one. You can do EEG concurrently with fMRI, concurrently with FNIRS, concurrently with brain stimulation, TMS, EEG, concurrently during surgery. So it really kind of fits in as a partner to multiple other measurement modalities, and in that way, really serves an important complementary role in giving us both unique information, but also concurrent information from that we can compare and understand better other types of measurements that we make. But the key thing really in terms of why do EEG, or I think really kind of goes top of the list, although it's largely a practical question, is that it's cheap and it's scalable, right? And that's kind of a major theme of what I want to talk about here in the first part of this presentation. So when I'm talking about cheap and scalable, there's really two levels to what we mean by that. The first is, let's say, compared to functional neuroimaging, like MRI. An MRI scanner the price tag for that is millions of dollars, right? And the price tag for running it is millions of dollars. And the price tag for running a study, once you have all that gear, is let's say $500 per subject, right? So these are large numbers, even for wealthy research institutes and wealthy groups. Research grade EEG is going to be something like a 20th of the cost, just a ballpark of research grade fMRI. So to actually kind of buy the system, the actual hardware, as I've got just listed here, and it varies a lot, but let's say $70,000 to 200K, depending on the system, etc. You'll need personnel. Obviously, you need personnel for everything, but really, there's nothing extra on top of that. Like once you have the gear, it's basically free to do EEG research. So that that opens up a lot more options. There's a lot more that you can do when you're working in that kind of price domain. However, that's still pretty high, right? That's a lot of money for anything really, but especially when there's tight budgets and a lot of competition and so on, or if you're a small scale enterprise. So that comes to the second part of the point about pricing and scalability, which is the right hand side of this slide here, which is what's generally called consumer grade EEG. And so these are, if you like, budget systems, but I think, and they are budget systems, right? So I've got a list of things like roughly in Canadian, and it's not that far off in US. We're talking about, say, like $500 mark for some systems, or maybe up to $2,000 for a really nice one. But generally that kind of territory. So, maybe a bit more than a Christmas present, but not too much more, I'd say. And obviously these are in many ways, they're like scaled down, pared down relative to research grade systems. But at the same time, you can do a lot of the same stuff. And there are even things that you could, like lots of things that you can do with consumer grade systems you can't do with research grade systems. And one of the reasons for that is because there's actually been a lot more technical innovation in this sector than there has been in this sector, let's say over the past 20 years. Like honestly, the hardware for research grade EEG hasn't changed a lot in 20 years. It probably hasn't changed a lot in 30 or even 40 years, to be honest. However, in the kind of consumer grade sector, there are a lot of innovations that have been introduced. A key one is wireless streaming, right? Which is kind of a requirement for the kind of form factor that you need when you're not in a lab, because what the kind of use case that generally you're aiming for, companies are aiming for in a consumer grade, or maybe if you like a mass market context, is people with less patience and less technical chops to deal with, and less time. Okay, so for all those reasons, like there's a lot of technological innovations that are often featured in these technologies, and just the fact that they're newer sometimes also helps. So sometimes the board, the electronics, the chipsets, the hardware of just microchips and circuit boards, like that actually evolves quite quickly. So like circuit board from the last 5 years with modern components is a lot better than equivalent thing from 15 years ago. So just the fact that something's been designed in the last 5 years automatically makes it in some ways better than something that was designed 15 years ago. But other things, like, as I was saying, wireless streaming, active electrodes, dry systems. So that's when you don't need to apply conductive gel to every channel. These are all things that as I said, they're kind of hardware innovations or hardware technological developments that they're kind of par for the course in consumer grade systems, and they give you a lot of added utility over and above just them being a pared down version of a research grade system. So this is an important type of technology, innovative type of technology. Obviously, from let's say from a neuroscience point of view, having a smaller number of sensors and having slightly less contact with the brain, which is what happens if you don't spend half an hour applying conductive gel. You say the signals are not as good. No point in questioning that. So from basic neuroscience point of view, yes, research grade systems are going to tell you more about the brain, but there's a lot that we can do in this space of consumer grade systems, and that's what we're interested in exploring with the project that I'm going to talk to you about now. Another aspect of what I was just describing is also, it's not just the systems, but it's also some of the associated, say, peripherals. So being able to run your experiment on a standard laptop with open source, relatively recently developed stimulus delivery and like software stack. Those are other things that are enabling factors for the type of things I'm going to talk to you about now that have really emerged in the last 5 years or so. Like, it wasn't the case 15 years ago that you could do these things. Even if you had the hardware, the software ecosystem wasn't available, but it is now. And we're trying to build it even further. So one way of summarizing some of the things I was saying just now, or making a compelling description of them, is to say, we like to think about this is say that what we're trying to do here is we're trying to democratize the cognitive neuroscience experiment. And honestly, photoshopping all these things on top of this like Delacroix masterpiece is one of the more fun things I've done over the last few days. But there's maybe two things to say about this. One is, obviously, I'm being a little bit frivolous, but at the same time, one of the things that we're trying to do here, we were generally interested in doing this, is moving the capacity to do certain types of research and non-research things, but like, things with EEG, moving that out of the strict domain of the, for want of a better word, like the well-funded fat cat research labs that have the resources and the facilities to do even just the research grade EEG that I had on my other slide. I mean, obviously the neuroimaging is another order of magnitude on top of that. But even having, spending $200,000 on doing some research EEG is outside of the scope of a lot of people who would otherwise like to do that. So we want to open things up to the world, open things up to different communities who have less financial resources but have interesting applications and interesting use cases. And those do extend beyond just cost. They include there's qualitatively different things that we can aspire to do in a low cost consumer grade context that are not just what you would do with the research system if the research system was cheaper. The obvious one is, well, one of the key reasons for that is some of the things I was describing just now about the other aspects of consumer grade systems, as in, they are intended for use cases where you don't spend half an hour or an hour doing setup. You want to just stick a device on your head and just get some brain data straight out. So that, as you can imagine, like, that means that you can do more things. You can do a lot more in the educational sector. You can do a lot more in the clinical sector. You can do more, let's say, just general outside the lab biosensing applications. Sometimes it's called quantified self. So there's actually quite a big hacker enthusiast community, the people who just like measuring their biosignals and knowing about themselves in that way. So you're facilitating that as well. So there's lots of benefits beyond just like saying doing a cheaper and slightly less good version of what you would do in the lab, and that adds to the motivation for what we're talking about here. I talked about the enabling factors just now. This will come back in a second. Good. So hopefully I've conveyed some of the motivation, maybe got you a little bit excited about understanding why we're interested in developing software that allows you to do cognitive neuroscience research with mobile EEG technologies. Now I'll go into a little bit more details on the nuts and bolts. So here's the link. This is the tool. The link there is to the documentation pages. There's also a GitHub repo which is where the code is that is linked to from the page there. I like my corruption of the Jupiter logo there. I hope they won't sue me for it. Interesting question. Let's see how it goes. Because it's an EEG, you see, it's got the nose and the little ears. That's what EEG topo plots look like. Okay, there are some other related projects. I'll maybe comment on the relationship between what we're doing and those as we go, but I do want to give a shout out to Kyle Matthewson's EEG EDU, which is a fully browser-based software library that lets you stream EEG data and lets you do some interesting experiments. Some, I think, useful overlap with what we're doing, but not exactly the same. And there's some cool stuff also coming recently from a company called Mind Effect, which we could probably talk about as well, but maybe later on. What are the aims? So 3 main aims really for what we're trying to do here. We want to be the go-to library for time-locked cognitive neuroscience and BCI mobile EEG experiments. We want to generate large open access data sets for the neuroscience and also for non-neuroscience community. And we want to promote innovation in research, in research, in clinical and in education, and what we could probably broadly call like grassroots or enthusiast communities. Now, 2 things to say in addition on this is on the first point. So on the one hand, so saying like we want to be the go-to library for this, it's both ambitious and not ambitious, in the sense that it's ambitious to say you want to be the go-to library for anything, and honestly, we don't care so much about that. We just want to make a useful tool, and it meets a need. And if other people make a better version of it, then all the better. The need is met better. But at the moment, there isn't anything else there, and that kind of speaks to the second point as well. Is on the one hand, it's ambitious, but on the other hand, it's not really that ambitious because there aren't any alternatives at the moment. There isn't a dominant software library that is doing the things that we're aiming to do with EEG notebooks. It's really kind of wide open space. So a second point about this project aims is what I'm highlighting here with the underlines. I'm saying time-locked cognitive neuroscience experiments and BCI mobile EEG experiments, and this is something that I do want to emphasize. So we're not trying to do everything under the sun with mobile EEG here, and in particular, just plugging in and recording data. That's not hard. I mean, there's a million mobile apps that'll do that for you. There's lots of good options for that. We're not per se trying to. We're using tools that allow you to record data, but it's not just about recording the data. It's about recording EEG data that is synchronized to the onset of stimuli that we're delivering in a cognitive neuroscience type paradigm. Okay, and that combination of the streaming and the like mobile data streaming and the recording and the synchronizing to triggers that are telling you when there are events happening in a stimulus stream, that's really what we're kind of focusing on, because there's additional technical features or challenges that need to be addressed in that, and that's our niche, if you like. Now, I've been talking about us and we and the project and so on, but really there needs to be some acknowledgements and a bit of a roll call before I go too far on this. And really the first and biggest of those needs to go to okay, sorry, I spelled the name wrong already. Alexandre Barachon, who came. So EEG Notebooks is basically by and large the brainchild of Alexandre. What he was doing quite a few years ago now was he was using the Muse EEG device and interested in doing some BCI machine learning, signal processing, and found that he needed to implement a bunch of things in order to achieve that. He needs to figure out how to stream a device. He needs to figure out how to cue stimuli and unify the information coming from the stimulus timing to the EEG stream, and needed some experiments to do that. Needed some principally visual and auditory experiments. So more than 50% of the groundwork for this project was laid down by Alexandre, and he's not actively involved in the EEG Notebooks library at the moment, but maybe he'll come back in the future. He is still actively involved in one of the dependencies, which is Muse LSL. and other interesting libraries. So Alexander is really kind of the first guy in the roll call. And then a few other people that I've highlighted here with some pictures. I mean, I'll read them out. So Hubert, who is with us today at BrainHack, Dano, who actually more or less kind of brought me into the project a few years ago. We spent some fun time in some cafes in Toronto in the summer around about 2016 with Muse EEGs on our head, trying to get some code down to get all this stuff working. Amanda Eason really helped out with a lot of things. Jason, who I'll also give another comment out in a bit for some streaming aspects. Kyle Mathewson from U Alberta does lots of awesome work, but also most recently gave us some cool new experiments. And most recently, in particular, Jaden, who's also here at BrainHack, or he's in the Gathertown a lot of the time. He's done a lot of key recent implementations. So this is an abbreviated list of contributors, but definitely the main folks. And I really want to extend acknowledgements to everyone else who's been contributing to the project, because there is a team effort. And one of the ways in which it's a team effort is not just through the individuals, but it's through the community group. So most of the people in the list on the previous slide, their relationship to the project, myself included, has been via this organization called NeurotechX, which, so I'm just giving you a little plug out here, is it's basically an international enthusiast community, but it's growing super quick. Like there's thousands of people who want to get involved and share knowledge and share expertise and build tools together. It's such a buzzing community, and so I really do want to give it a shout out. And it's really helped for bringing in enthusiastic developers and contributors and getting some attention and so on. So this has been really kind of a central piece of the picture in terms of the EEG Notebooks project all along the way. So shout out to NeurotechX and the people who've helped develop that over the years and continue to do so. Now let's get into the nuts and bolts of EEG Notebooks. So basically what we're doing is three things. We're taking data from an EEG device that's streaming wirelessly, and that component is, we basically have two principal libraries that we're using for the streaming. One is this tool called Muse LSL, which, as I mentioned, was developed by Alexandre a while back. And actually, the kind of history of things is that initially there was the Muse LSL library that had all of the EEG Notebooks components in it, and then around about 2016, it was mainly Dano Morrison. He basically pushed the two libraries apart so that Muse LSL was focusing just on the streaming component, and EEG Notebooks focusing on the everything else. So Muse LSL is now a dedicated streaming library, and it deals with streaming data from a Muse EEG, such as one of these. And the other library that we're now depending on, and this is one of the major developments that's come in the last is it a year, maybe more than a year now, but pretty recently, is a great library called BrainFlow that's developed by a fellow named Andrei Parfenov and a number of other colleagues. And BrainFlow basically is it allows you to stream signals from a large number of devices with some nice low-level kind of C implemented streaming functionality. So we're using BrainFlow to basically expand the device support options, which I'll talk about on the next slide. So first stage is get your EEG data. And then second stage, I mean, we're doing this concurrently, but the second component is run an experiment. Okay. So I've mentioned cognitive neuroscience experiments before. By the way, you can take one or other perspective or both perspectives on what we're doing here. My perspective, kind of a function of my background, really, is that we're doing cognitive neuroscience here, but some of the kind of neuroengineering community tends to think about things more in terms of brain-computer interfaces. There's obviously no sharp line between those two, although there might be a continuum. But yeah, so stimulus delivery, call it cognitive neuroscience or call it BCI, whatever you like. What we're doing is we're presenting stimuli, visual and auditory stimuli, and doing that in an organized fashion that has this timing information that I've talked about a few times. And the principal library, well, the only library at the moment that we're using for that, although we're looking to expand in one or two areas, is this great tool called PsychoPy, which is quickly becoming the go-to tool for designing and running stimulus delivery-based experiments, because it does a great job with the timing, and it's also open source. So the third component then, we've got the stimulus delivery, we've got the EEG. I'll show you a little bit more on both of those shortly, is organizing the data, organizing and analyzing the data. And there we're mostly depending on M&E. M&E is the really kind of one of the flagship libraries, I would say, in open source neuroimaging, particularly the Python-based neuroimaging community, not for MRI, but for EEG and MEG, and also recently, in addition, FNIRS. And M&E is great, basically. So M&E is the right way to organize and analyze your data if you're working with EEG data, as far as I'm concerned. So that's how we're doing things. And this is the package. This is what EEG Notebooks is. It's the combination of these three things in the service of doing cognitive neuroscience experiments. In terms of devices, so as I mentioned earlier on, initially, until relatively recently, the focus has been around Muse EEG devices, these nifty little headbands. There's also now the Muse S, which I've been playing around with recently, that's designed for sleep data. This is more like a sports band. This is a really nice piece of kit. They have like fabric electrodes and stuff, little detachable battery thing on the front. So Muse devices are the main type of mobile EEG device that we've been focusing on so far. But as I mentioned, as of relatively recently now, thanks to BrainFlow, and particularly thanks to Andre and Jaden, who developed BrainFlow, and then Jaden, who took BrainFlow and implemented it in EEG Notebooks, we now have a wide range of device support. The main focus in this area has been OpenBCI, and we've actually been working with some great folks at OpenBCI to develop the tools, and also to get the message out and do some interesting research studies. This is kind of on the horizon. So OpenBCI is the main kind of second device that's supported, let's say. But in virtue of BrainFlow's broad device support, we now have the new. This is a pretty new, pretty cool company called Neurosity that they have some really nice mobile EEG systems, slightly different to, say, the Muse, because they're trying to get channels at the back of the head, whereas Muse is focusing on the front. Other ones, BrainBit, G-Tech, which is an established like research grade EEG company, but they have some nice mobile devices like the Unicorn as well. And actually the list goes on. So really now there's a very exciting new phase where we're becoming a kind of universal mobile EEG cognitive neuroscience platform, mainly thanks to the functionality that's coming through BrainFlow. This is very early days on this, I should say. Like, we haven't done very much testing with any of these, but we know that we can connect, we know we can get signal, and we're definitely looking for people to chip in and try things out and help us get some pilot data and see what we can do. So what I just described, this is getting into the code a little bit. We have a generic EEG device that is kind of funnels to either the Muse LSL or the BrainFlow backend, depending on what you tell it your device is. And then a key thing is the experiments. So we have a library of experiments. Some of these have been around for a while. Some of them have been around. Some of them are relatively newer. There's a few more that are in the pipes. The go-to for a lot of what we've been doing is this one, the Visual M170. So this is a faces versus houses visual presentation experiment that I'll actually show you in a second. but the key thing is really we're expanding this. It's relatively easy to expand this, and this is pretty exciting. So we're getting together a library of experiments in one place that's really implemented and like runnable in a fairly uniform fashion. And there's not actually much out there that does that, even for just straight up visual or sensory psychopy-based psychology experiments. I mean, not in the way that we're doing it here. There's Pavlovia, which kind of, will run in the browser, but it's doing something slightly different. But so the library is expanding, and that's great. And we're definitely looking to expand the library further. Here's just an example in case all of what I've said is not particularly clear to you. I have to go off the laser pointer to play this little video. Oh, come on, play the video. I tested this out. I'd really like to have that go. I'm actually going to exit the show briefly and then see if I can play this for you from there. Okay, I'll just zoom in so you can see this a little bit better. So this is me in the terminal, and the reason I'm showing you this is so that you have an idea of what I'm talking about. If it's not really clear, I've been talking about visual rapid serial visual presentation experiments and so on. So I fire up my terminal, I start Python, I import one of the experiments. It's just as simple as just doing a straight Python import, and then I just run this command, which is basically run experiment called present, and I tell it how long I want to run it for. In this case, I'm just going to do it for a very brief period of time because I just want to do a demo, so just 20 seconds, and then it will fire up. Well, we get a instruction set first, and then we're going to get a bunch of pictures. So the participant is staring at the screen with the EEG on their head at this point, and what we're getting is brain responses to faces and brain responses to houses. Now, people who are familiar, maybe have a psychology, cognitive neuroscience background, they'll know that that's a big thing in cognitive neuroscience. Faces versus houses. Oh my God, especially in fMRI actually, but also in EEG and MEG. The reason is interesting is because it's to do with it's to do with expertise. It's to do with specialization in the visual system. It's to do with magnocellular versus parvocellular pathways and kind of retinal versus non-retinal information processing streams. Don't need to go into any of that, but anyway, faces and houses, yeah, big thing in visual cognitive neuroscience. So you run the experiment as I just showed, just in this little diagram here. Have this guy on your head, and it gets something like this. So this is a average response over a couple of hundred trials. And what we're looking at here then is two things. We've got our red line, which is the averaged trial averaged visual response to the house stimuli, and the green line, which is the trial averaged visual response to the face stimuli, and then the black line is the difference between the two. And what we see here, which is cool, is an N170 response. Now, the reason why the N170 is a face selective visual response that occurs at roughly 170 milliseconds. That's why it's got the name. We're seeing a face selective visual response. It's face selective in the sense that there's a big difference between the green and the red line at this point. So that's why the black line is showing this inflection here. But it's not at 170 milliseconds. It's later. It's just over 200 milliseconds. So why is that? Well, two reasons for that. One is M170 is a ballpark, right? It's not everyone's visual face selective response isn't bang on 170 milliseconds. But the second reason is because we know that there is a little bit of a lag in Bluetooth wireless streaming. So this is a little bit of a weakness in the like hardware stack that we have that we're using here, is that there's going to be delays that are introduced that are somewhat systematic, like fairly systematic, that we just have to account for in our interpretations on. So we know that this response is actually somewhere earlier. It's somewhere the calculations for the Bluetooth lags have tended to hit a ballpark of 20 to 30 milliseconds. So that actually pulls us back to roughly where the M170 response should be. But again, this is like a bit of a weakness in the system, that in the approach. It's not an issue with Wi-Fi based streaming, but it is an issue with Bluetooth based streaming, which is what most wireless devices do. But otherwise, it's really an interpretation thing, because it's consistent enough for you to just say, okay, well, we know that this is coming from a little bit earlier than the 200 milliseconds that we're seeing it. So good. So we have a similar experiment with a visual oddball paradigm. In this case, it's cats versus dogs. Quite a fun experiment, this one, to do, because you get just pictures of cute cats and dogs coming up. This is an interesting one. This is steady state visual evoked potential. I don't have the stimulus here, but what it is a flashing, what we call grating. So it's like a black and white or black and gray lines that are flickering at a specific frequency. And the frequency that they're flickering at, there's actually two stimulation frequencies, one 20 hertz and one 30 hertz. So the plot that I'm showing here, this isn't a trial averaged ERP response. This is a power spectrum. This is the power spectrum that you're seeing in the temporal channels and in a occipital channel that you can add to the Muse device if you plug it into the little spare socket at the back. And we see exactly what we expect to see, which is a peak at 20 hertz when we're doing the 20 hertz flicker, and a peak at 30 hertz when we're doing the 30 hertz flicker. So you're introducing basically an oscillatory brain response, an oscillatory rhythm, by presenting a oscillatory visual stimulus, and you can actually essentially inject the frequency of the visual stimulus into the brain at the locations that we're recording from. That's what steady state paradigms do. Steady state visual evoked potential and auditory evoked potential paradigms do. So that's cool. It's also interesting because that's a visual gamma flickering has been earmarked as a potential cure for dementia and Alzheimer's, just as a side note. So maybe we should be doing more with this. But it's been a bit of a boon for SSVEP paradigms, those results from mice that showed that you could flicker a stimulus at a gamma frequency and it would reduce tau protein levels in Alzheimer's mice models. Side note, really. Okay, decoding or machine learning. So this is something that we're doing, and actually there's some cool projects on at this BrainHack, which is applying machine learning techniques to do classification of, for example, the faces and the houses stimulus sets or trial sets. And this is an area that we're interested in. We're definitely interested in developing, and one of the reasons why we're interested in developing it is because this is something that people can contribute to without having to have any of the hardware or do any streaming and do any data collection. It's really like, we have the data, we want people to do interesting ML things and statistical analysis things with the data that we have for the conditions and the experiments that we have. So we're kind of offering that out to people who are interested in doing some development and some data exploration in that area. So definitely get in touch if that's something that might be of interest to you. I'm going to quickly roll through a few other newer features, maybe more for people who've maybe seen the tool before, but not so recently. So we now have a command line interface, which really functions just like a standalone command line program. So you can just type in EEGNB, run EXP, and you can just run experiments anywhere in the command line, no Python code or anything, just type it in and the library kicks off. We have dataset fetches, so we're not keeping data in the GitHub repo anymore, which was how it was organized previously. is inspired from the functionality in tools like NiLearn. So we implemented NiLearn type data fetchers. We implemented Sphinx Gallery documentation, again, following folks like the NiLearn and the M&E folks, and makes things pretty and also makes things easier to manage in terms of version control and CI. So that's been a nice progression in the kind of computational quality of what we're doing as within the library. And another thing is actually going back a little bit now, but this also was what brought me into the project to a large extent, is educational initiatives. So a few years ago now, we ran a project in some schools in Toronto called NeuroBright. That was basically a bunch of us volunteers at Baycrest, which is where I was working at the time. We were going into 3 high schools, and we spent a whole term working with high school kids, like final and penultimate year high school kids, on a Muse-EEG based cognitive neuroscience science project. They designed an experiment, they ran an experiment, looked at the data, they presented their results in a little conference workshop thing that we organized, and they did something that's I think really unique but possible for at the high school level and at the undergraduate level. So this was something that I was speaking to earlier, is there's a lot of scope for using these tools that are really doing, quote, real science, right? The visual face house responses, like that's something that card carrying perceptual psychophysics cognitive neuroscientists do. But you can definitely do that in a high school. You could do that in an undergraduate science class. You could run the experiments at least very easily with large numbers of people with the hardware that I've been talking about, and with some of the software that we've been developing. And that's one of our kind of target areas amongst others. So we did NeuroBright in Toronto. There was also a similar project running in New York called Brainwaves. And of course, at BrainHack, we've done 1 or 2 BrainHacks now focusing around EEG notebooks, and we'll continue to do that. And that's partly an educational initiative as well as a development initiative. So final comments then is the future. What does the future hold for this project? Well, we want more. We want more experiments. We want to have support for more devices. We really want people who are using more devices, different devices than the Muse BCI, to get in touch and try out the stuff and help us develop the support and see if you can do it more and better, say with the Neurosity Notion than with an Open BCI. These are things that we're interested in knowing. And more research. So there's a few developing research projects that are building on the tool now. So we're really kind of branching into doing some straight up academic research. And large-scale data collection and open science. So this is one of the things that we really want to start paving the way for, developing the procedures and the kind of framework for doing citizen science with these tools, but in a way that is ethically sound, which is a key component of it, but is distributed in the sense that we're not going around with our laptop and our Muse EEG and handing it to people and get them to sit down for an hour in their in a room in a library, whatever. We want people to do it in their homes with their devices on their laptops, with us not having anything to do with them. And we want a mechanism for using that resource and capitalizing on that resource, and also the enthusiasm and the kind of brain power that the international research and non-research neurotech and open science communities have to offer, and that is a serious resource. We want to harness that resource in the service of our broad objectives. So that's something that we're working on, as I said, the kind of mechanisms and the processes for. One of those is going to be a series or a series of both research studies and data analysis competitions that that we're calling the Neurotech Challenge Series. We're organizing this in collaboration with OpenBCI and NeurotechX. We've put the date back a little bit, so we were a bit optimistic with the start time, but this is happening soon, and this is going to be trying to do what I just described. It's getting people who you have a device, right, and which is not everyone, but if you do have one, or if you're interested in going and getting one to do this, then you can do some cognitive neuroscience BCI research with us in the comfort of your own home and contribute to some research activities there. And also, if you don't have a device, then we'll have some data analysis competitions and also research studies that we'll be looking to do that kind of leverage the data that we're looking to collect in this context. And to wrap it all up, this is a version of what I was presenting to you, proposing to you as a democratizing influence in the domain of cognitive neuroscience. Really opening things up to a broader audience and a more diverse group of people to contribute to human flourishing in this small little way. Cool. So that's it for my intro session. I'll field some questions from you guys now, and then we'll take a break, as I said, and then we'll do our live demo and run through after that. So I'm going to flash back to the Crowdcast and see what we've got in the Q&A. And I'll stop screen sharing as well. Okay, so no burning questions in the Q&A box right now. I'll hang on here for a minute or two, see if any questions come in the thread, but I'll also propose a time for us to reconvene for the second part. And the time I'm going to propose is 2:40. Yeah, 2:40 EST. It's just a quick session, so it's going to be a 15-minute job, and that'll take us up to the end of the slot. Any burning questions from people? And clearly, I've completely answered everything that people would have possibly been unsure about, and done a jolly good job of conveying all of the information. So you don't need any more help. Okay, all good.
I actually had one question.
Oh, Kevin, you have a question? Yeah, of course, you can ask a question.
So I'm not sure if I missed it specifically, but how do you, like, conventionally with a normal EEG cap, you have different nodes that are pretty standardized in like the literature that you like when they discuss it. How do you kind of go about defining a specific area and knowing that everyone's going to kind of wear it a little bit differently, so there's no way to make sure it's like exactly the area you want to measure? Yeah,
well, it's a good question. I mean, that's also the case for a standard, say, research grade 1020 cap, in the sense that you don't know exactly where you never know exactly where the channels are unless you've done head digitization, which is what you would do if you're doing like a fully neuroimaging integrated EEG research. You would take an MRI scan of and then basically find the shape of someone's head from a surface from an MRI scan, the head and their face, and then mark the fiducial points on the MRI scan, and then mark the same fiducial points on we call like a head digitizer on the EEG, and then you know where the fiducials are in the EEG in relation to the MRI, and so on and so forth. So like that's the only way you really know where the channels are in a meaningful sense, and the meaningful sense in that case is in relation to someone's brain. But more like to your specific question, yeah. So with the Muse device, the nominal channel locations are FP9, FP10, TP9, TP10. I think I got the numbers. Did I get the FP numbers right? Anyway, frontal and temporal channels, and the temporal ones are kind of approximate, so they're not far off where it would generally you generally get that location with the 1020 cap, but they're not like bang on. So there's a little bit of hand waving, if you like, in terms of the specific spatial location, depending on what you have. So another one would be the OpenBCI Ultra Cortex, which is more or less a standard 1020 system, and it's kind of as close as an EEG cap, a standard EEG cap would be. So there are some consumer grade devices that are like a little bit more accurate or a little bit more standardized in terms of the locations, but it is it is like a bit of a, as I said, a kind of fuzzy area in terms of the signal you're getting. And you just have to appreciate this, is that, you don't know that it's exactly TP9, but honestly, what is exactly TP9, right? So the EEG signals, they're very kind of, they spread a lot. So with high density systems, you can discriminate more spatial, you can get more kind of spatial resolution, particularly with source analysis. But if you're going to get a signal at TP9, you're going to get the same signal an inch north of TP9. Like that's just how EEG works. There's not going to be like a massive difference with a little bit of blurriness in the spatial locations. So I guess that's the simple answer to the question there.
Sorry, before I ask, I'm going to ask all my questions selfishly before I ask the other ones posted here. But is that part of the reason that you can settle on those specific locations, I guess, just due to the attributable to what you want to measure, or is it more about like the cortical geometry, especially without conductive gel, is a lot better in these particular regions to read in data?
Yeah, it's a bit of both, basically, and that's not our decision. That's the device manufacturer's decision, right? And then in their designs, but that's one consideration in addition to other considerations like form factor and like how to stick it on your head and so on. But a TP9 is a good place to record EEG. Like you get good strong responses from there in any setup, in a research grade setup, in any mobile device. It's just you get strong brain responses around the temporoparietal junction. You get strong brain responses around the CZ area, so like central motor cortex, like near the motor cortex and near the somatosensory cortex. Like that's a good general spot for EEG signals as well, and also tends to have less motor artifacts, and occipital as well. It's a good place to get certain types of, especially obviously visual signals. So there are some places that just give you good EEG, and in the case of the design of like this device, for example, like obviously they know that they get good EEG around the temporoparietal area. That's why they've designed it to have channels there.
Right. Cool. Thank you. So there's two questions here. First is from Manuel, who asks, are there plans to include neurofeedback tasks?
Yeah. Hi, Manuel. So the short answer is not immediately, but a slightly kind of more elaborated version of that is, you might have seen the new stuff from Mind Effect. I actually had that quickly on a slide there, but that's a real-time. It's not exactly neurofeedback, but it does let you do real-time BCI kind of flicker-based visual field device control. And so again, not neurofeedback, but more towards the area of a real-time signal, which is one of the things that you need for neurofeedback. And we're interested in borrowing some of the functionality from that library for the stimulus presentation. We're kind of on the fence at the moment as to whether to try to go towards real time. I mean, real time would be great, but as I said, one of the things that we're focusing on these ERP paradigm type experiments, and you see the ERP when you average, right? So having like real-time information is useful when you want to do something in real time. But if you're looking to kind of take multiple trials and find what the trial average response is, then it's less clear that real time is, super relevant for that. But yeah, these are like open questions that we're having amongst the group, is like what extent we want to incorporate neurofeedback and want to incorporate real-time functionality into the library. And maybe we will in the near future. It's definite. And if you want to come and do that, then you can definitely come and talk to us about it.
Second question is from Vera. Are you planning to switch completely to BrainFlow devices, or do you want to stay with both Muse and BrainFlow devices?
Yeah, that's a very good question. We would love for BrainFlow to support Muse, and then we would only need to support BrainFlow. My understanding is BrainFlow don't intend to support Muse, and I don't know what the reasoning around that is, but we're definitely not going to drop support for Muse devices. But if there is a better, as I said, like if BrainFlow have a good support option for Muse, then that would make our kind of maintenance job easier. But as I said, the history of things is really, everything was based on Muse and everything was based on Muse LSL. So it will be a pretty big, pretty big shift to kind of drop that out. But yeah, depends on the Muse support in BrainFlow basically, and talk to Andre about that. And if you can persuade him to put Muse in BrainFlow, then I'd be happy.
Okay, cool. You said you want to meet back at what time?
Yeah. So let's take a break and let's meet back in 5 minutes. So we'll do 2:40 EST, and we'll do a relatively short live demo. Okay.
I'm just going to keep the stream going here then, and I'll cut out our video for the time being.
Okay, good.
Great. All right.
Right. Thanks, guys.
I like the look.
Yeah, this is. I got to take the hit, you know. If I'm gonna do some proper tutorial, then I'm gonna. But the thing is, like with the headphones, like I probably should have a different pair of headphones because they interrupt a bit. Anyway, yeah, I think we're good for now. Ready to go.
Okay, cool. Well, here, you got the floor.
Yeah. Oh, we're live already. Okay.
Yeah.
Hi, guys. That was me thinking we were in the green room still. Cool. No worries. Welcome back, everyone. Yeah, so slight change from me. Can you spot the difference? Can you see it? Can you see it? Yeah, that's right. My right mustache is slightly curly now. I put on an EEG, the Muse EEG device. So I'm going to do probably 3 things with you guys in the next 10 minutes, just to give you a flavor for how these things go. So first thing is put on said EEG device. I won't go into the ins and outs of that, but that's another discussion as to like what the optimal way of putting a Muse on your head, especially if you have a little bit of hair, to get it out of the way. But in any case, pop it on your head, and then so now I'm going to share my screen and show you a few things. Right, so hopefully, just double check that you're seeing my screen. Yep, you are. Okay, so I just ran this to move things quickly, but you can imagine for yourself. So I've just pulled up my Anaconda terminal and activated my environment, and then this is it's not an EEG Notebooks, this is a Muse LSL command, but this is going to launch a live viewer. Now, one other thing actually before we look at that is for Windows users, I'm using a tool here called BlueMuse, which is it's kind of a third-party library that we need to use to engage the EEG stream. So basically, I pull up BlueMuse, it finds the device. You can actually see it's detected this device. That's the one that I've got on my head. I tell it to start streaming, and it kicks open this little tab here, and that tells you that it's streaming data from the 3 sensor types coming from the Muse, the EEG, accelerometer, and the gyroscope. We're just looking at the EEG here. And so I have my stream going. The next thing to do is I want to know that I've got a decent signal quality. So here I'm going to want to launch the viewer, as just said. So just run this command to launch the viewer, and then we'll get a live stream here. Now, what we've got here is 4 channels, the 2 temporal channels, TP9 and TP10, and 2 frontal channels. This is 7 and 8. I got the numbers wrong, AF7 and AF8. And what we're looking for here, so when you're doing this, the number you want to aim for, these numbers on the right-hand side, these are the variants of the signal. And this is our kind of time series-based substitute for what you would normally use impedance for if you were doing a kind of in the lab research EEG study. But basically what you want the numbers on the right hand side to be as low as possible, and ideally be in the range of around about 10 or under. So you can see that I've got variance levels below in kind of good numbers. I mean, I'm speaking at the moment, right? So these are kind of going up and down, but when I'm still, you can hopefully see that these are in the ballpark of 10 for the 2 frontal channels. The temporal ones are a mess. Now, the temporal ones are a mess because I'm wearing these headphones. So I'm actually going to try actually, no one's speaking to me right now, so maybe I'll just take the headphones off for a bit and hope that there aren't like major objections coming. So I think these are going to improve a fair bit now if I stay still for a second. Yeah, famous last words. I think because I've had this running for a while, there's a bit of a lag in the stream, and also because my computer's doing multiple things at the moment. So what I'm going to do is I'm going to kill this and then restart it, and then we'll check back at it in a second and see if that temporal signal's got a bit better. It doesn't matter so much for what we're doing right now, but interesting and also essential for essential when you're actually running these experiments that we're going to look at, because you need to check that you've got good EEG signal before you start recording. Otherwise, you're going to have bad data. Right? Now, so in addition to doing the visualizer, I showed you in a little video running the experiment. So the process for that, if we just kind of quickly look at the terminal I had, but I won't rerun it. It was just import experiment and then run experiment. Now, doing that on its own, that just gives you the stimulus presentation, but it doesn't do the data collection. So it's a slightly different, slightly more involved command to do the concurrent stimulus presentation and the data collection, which I'm not going to do right now because I want to actually focus on showing you something else. But in a nutshell, what I would do is I would import. Actually, I'll just show you how it looks in the documentation, and then we'll comment on that, and then come back to what I want to show you next. So the syntax for running an experiment looks like so. If you're doing it in live Python, we also have the command line interface where it's just a single line that I would just run in the Anaconda terminal. But you can also run it in Python, basically running the commands that are called from the scripts that the command line interface calls. And it look like so, kind of import two main things really. One is the EEG device class, and the other thing is the experiment class. So we've got something about our EEG streaming and something about our experiment. And then we need to initiate the EEG device, just this line here, and I need to tell it what I'm using. In this case, be a Muse or a Muse 2016. And then, as I just said, so then I'd run the same command that I showed you with does the stimulus presentation, but in addition to telling it how long I want to do the stimulus presentation for, which is what this record duration is, I'd also hand the EEG device class to the present command. And then what goes on under the hood then is it runs the experiment, but it also pushes samples to the process that's saving things to file from the EEG at the same time as pushing the triggers from the visual presentation. Good. So that's kind of it for the running things component. Let's just see how okay, that didn't run yet because I needed to a little bit longer. Let's give this a second. Let's see if I have a slightly better signal in my temporal channels right now. Yes, I do. Okay, so that was just to kind of convince you that I was getting a bad signal because I had those headphones on, and that would be important because especially if I was doing an auditory experiment, and you need to be aware of things like that. You can see the variances flashing up on the right hand side. I could also, to show this is real time, just do some quick teeth gnashing. Right, that's quite a handy way of checking that your real-time viewer is actually in real time. Because if you do your teeth gnashing and then you only see that noisy component in the signal like 10 seconds later, then you know that you've got a bit of a lag in your viewer, and it's hard to know that otherwise. Okay, good. So we have our EEG streaming. We've taken a look at the signal quality. We've looked at how to run a visual experiment. Now let's quickly just look at what we do with the data. Put my headphones back on now, in case someone's shouting at me that they need something. I can hear you now if you're shouting at me that you need something. And so we're now just going to kind of take a look at one of the notebooks that's in the documentation. For reference, what the file that we're looking at can be found here. If you go to the documentation page and just the tab on the right hand side, and it's the load and visualize data. So we're just going to go over that and I'll give a bit of commentary on it. This is using data that is in the repo or the example data that's in the repo. I'm not using data that I recorded just now because I didn't do the experiment just now, but you can do this. You can do this. So you can do this right now with this data. You can download the example data and run this. And obviously, if you have a device, then you can make your own recordings and you can analyze your own data. And it's just which files you're pointing it to, the difference between those two. So first thing we're going to do is load in the things that we want to do. And as I said before, our main tool for analysis and data organization is M&E. So we're using some tools for M&E out right there. So first we want to load some data. So this command is finding some data that's in a default location. So the location that we've decided to put EEG data by default it's following the same as M&E and DiPy and the other Python kind of neuroimaging community packages, is we stick stuff in a hidden folder in home. Okay, so I'm in Windows, so which means that things are weird, but don't worry, this resolves to an okay path. But so it's in a hidden folder, so I'm in like my home directory and then .eegmb and then data. So that's where the example data will get downloaded to, and that's where your data will get recorded to if and when you do record data, if you record some data. And where it'll go after that is, just quickly look, what's in this folder. So, I need to put my glob. No. So the format for the data sets is experiment, then site, then subject, then session. Okay, and what that means is, so in this case, if I do visual M170, so that would be the experiment. Within visual M170, I have a few folders here. The key one is, well, the key two are local and EEG NB examples. These are the ones that you would see, but what you would see is this one, which is where the example data will get downloaded to, and this one, which is where any recordings that you make will be saved to by default. And then if we look inside one of those now, just to finish what I was saying just now. So what's going on inside this EEG examples folder? Okay, device as well. I forgot about device in the folder path, but let's have that as well. Spelled that wrong. Subject. Okay. After subject, we've got session, and then I won't continue. But if we go down, then we have subject. So each subject, we can have multiple sessions. Within each session, we can have multiple recordings, and that would be, for example, so I would say a session is, I sit down and I'm going to do 10 minutes or half an hour of EEG, but I'm going to do it in 2-minute blocks. I'm going to do, say, record 2 minutes of faces versus houses, brain responses, and if I tell EEG Notebooks that they're all the same session, then I'll have several 2-minute recordings all saved within, let's say, subject 1, session 1 folder. So that's how their data is organized. We're loading, so now we're going to load some data in. We have this fetch dataset function. This will download the data. I mentioned the data fetchers earlier. This will pull the data from our cloud storage location for the example data. If you don't have it already, I already do, so it doesn't need to do that here. And then here, this is just loading in subject 1, session 1 for the device Muse 2016 and the EEG NB examples site and the visual M170 dataset. So what we get is an M&E raw object. So this is, people who've used M&E before would be familiar with this, but it's really just the most generic data container object that M&E offers to you. And mainly it stores, well, it stores a few pieces of information. It knows about channels. in this case. So we've kind of organized it so it knows what the names of the EEG, the Muse EEG channels here. It has the data and a few other small pieces of information. The STIM channel here, this is the trigger channel. So this is what's coming from PsychoPy that's telling us what it actually is. If you actually look at the raw files, which are CSV files, the raw files are being recorded. You'll have a line. Basically, you have a row for every time point, and then in a column for each of these. And then in the column for the STIM, you'll have zeros and then a 1 whenever there's an event. Or sorry, a 1 or a 2 whenever there's an event. The event being the start of a trial. And so with that information, and that's how EEG and MEG data are organized using trigger channels like that. Then we can go to, I just need to run these 2 cells. We can go to, ignore the power spectra for a second. This point where, again, we're using an M&E functionality, which is epochs. So we get an M&E epochs object, but we've done a little bit more in it now. We've actually applied. What this is doing is it's cutting up the raw stream into trials, and it's giving us now a list of trials that are kind of cut up and, if you like, aligned on top of each other with a start and end time, which we've set to -100 and +800 milliseconds. And also we've got a little important little component here, which is a rejection threshold. So if this number, which is the signal variance, is too high, then we're just going to reject it. And this is a mechanism for making sure that we don't have really noisy data in the epochs when we. So you can see in this case we've got a sample drop of 4%. If I change this now, so let's say to e to -5, then we'd have a sample drop of 0 because we've made it a less stringent threshold. If I make it to the -3. Then we're going to have more drop because it's a more stringent threshold. I get that wrong. 0. Okay, who's it? Up to -6. That's a more stringent threshold. Okay, I want to see more drop. Okay, now we lost everything. We'll take it down to the -8. Okay, so we'll just leave that as it was. We've got now around about 500 per condition. And this is the final thing I want to talk about. So then we have this plot conditions function here that's going to show us the data, and this is going to give us the averaged responses plus minus some variance, standard deviation that you can see in a faint colored background there for our different conditions. Great, yeah. So that's it for the hands-on tutorial. Most of the second part of this you can just run through yourself. You just install the library, download this notebook, and then you'll be able to do everything I did for the last 5-10 minutes. The streaming component and the data collection component, obviously you need a device for, and we are here to help you with anything you want to do on both of those fronts. And I hope you enjoyed the talk. So I see we're at 3 o'clock. Any burning questions from the audience? No, I don't see any additional ones from the ones we had just now. So I think we can wrap up this session and just about stay on time with our program.
Yeah.
Thanks a lot, guys.
Thank you. I feel like I'm just on here to like clap and say, okay, we're done now. Okay, we're starting now.
Yeah, I've hosted enough Crowdcast sessions that I'm just like taking the ropes.
From Manuel again.
Can it be run from Colab, from Google Colab? Yes, you can do the analysis stuff, but you can't do the streaming. So because the streaming, you need to be running Python on your device. But in Google Colab, yeah, everything should run. You'll just have to do a few extra installation commands. Okay.
Sorry, I didn't understand.
No, that was my Google right there.
All right, great.
Thanks a lot, guys. See you in the gather.
Thanks.